Hello, um, good afternoon, and welcome to session 4, where we'll have two fantastic presentations, and moreover, another excellent example of the transformative power of bringing people together, bringing excellence and intersectorial research. And first, let me introduce Dr. Melanie. She's a member of the Coalition for Epidemic Preparedness Innovations, known as CEPI, where she's the Director of Vaccine Research and Development and leads the technical teams for vaccine development, enabling science projects funded by CEPI, and also the leader of R&D and the manufacturing work system that, for COVAX. Melanie is a physician specialized in virology for over 20 years of experience in the development and licensure of vaccines for the developed and developing world. Over the years, she has contributed to many vaccines, including seasonal influenza, pandemic influenza, pediatric combinations, and many more. And today, she's going to talk about new vaccines for a safer world. Thank you, Melanie, for being here.
Thank you very much. I just want to see if I can advance my slides, having minor technical issues. Unfortunately, I cannot advance the slides. Okay, what I will do is ask. There we go. Okay. We've overcome the technical problem. So thank you for the opportunity to come and speak with you all today. So indeed, I will speak a little bit of what CEPI has been doing over the COVID-19 pandemic, and a little bit of forward thinking in terms of how we should potentially be better prepared for pandemics in the future. So first of all, on the next slide, if you can really maybe just start off by introducing CEPI to you. So CEPI is a coalition of epidemic preparedness innovations that was formed following the Ebola epidemic in 2014-2016 in West Africa as a global organization that would help the world be better prepared for epidemics and pandemics in the future. And the way that we are working is really through partnerships with academia, with industry, with the regulators, governments, etc., in order to accelerate vaccine development for emerging infectious diseases. So if we move to the next slide. To start off with, so with CEPI's mission of accelerating vaccine development, as we set out in 2017, we had objectives to develop vaccines for a set of priority pathogens, which were on the WHO R&D blueprint list of viruses with epidemic or pandemic potential. So we have been partnering with vaccine developers for developing MERS, Lassa, Nipah, chikungunya, and Rift Valley fever vaccines. You can see here we are funding and supporting a number of different vaccine candidates. Then in addition to that, we were also working in advance of the pandemic on partnering with developers of rapid response platforms to prepare for disease X. So mRNA as an example of a technology and partnering. And obviously, probably before we all really wanted it to, then our disease X came with COVID-19. And where we are today is that we have invested in 14 vaccine candidates, of which 11 are still active. And I'll tell you a little bit more about those on the next slide. And perhaps before going there, just to say a few comments in terms of CEPI's work, as well as funding development of vaccines per se. We actually feel it's vitally important to look at enabling sciences to enable those projects to move forward. So some of the other activities that we have funded and partnered with are around building networks of animal model partnerships, building networks of centralized laboratories, in particular with COVID-19. And with that, with the laboratory network we have, we do offer to all developers the opportunity to send samples to the laboratories to have them tested against COVID-19. developing assays and standards and harmonizing assays is something that is critical to us to allow us to have good quality assays and comparability across different candidates. Also been working on variants and looking at the biological relevance on variants. And we also have other types of enabling science projects, such as epidemiology, and we have a large epidemiology study in West Africa, as well as other activities like community participation projects. But if you then move to the next slide, and the next. So we'll move into a little bit more detail in terms of how CEPI has responded to the pandemic. And I think it's fair to say that very early 2020, when we were seeing this pneumonia virus emerge from Wuhan in China, we felt we needed to act very quickly. And the way that we acted was really building on the partnerships that we already had. I've mentioned we were working with partners on developing MERS vaccines, not just for MERS as a disease, but also we were all very aware of coronaviruses as a potential pandemic threat. And together, marrying that with our work on rapid response platforms for disease X, we were able to hit the ground running and announce the first 3 partnerships in terms of vaccine development within 15 days of the sequence being available. And then what you'll see on the next slide is how, obviously, as we saw the pandemic continue to develop, those first 3 partnerships were at a time when there were only 581 cases, but it very soon became evident that we really needed to build a vaccine development partnerships to meet the pandemic. So we actually built one of the world's largest portfolio of vaccines, funding development of a broad range of vaccine candidates. And this is really built on the context of 3 major pieces. The first being speed of development. So looking at funding rapid response platforms. And as we knew at the time, they were not well developed and validated platforms. So although they held promise, they were high risk potential candidates. The other particular area is scale. And here we're talking about scale of manufacturing. So as well as developing the rapid response platforms, looking at developing vaccines that were easily scalable, so scaling up and scaling out to be able to fully utilize the global manufacturing capacity. And then the third key element here is access. So with our partnerships, within the contracts, we have first rights for vaccines that have been developed from our funding to be used in a global fair allocation system, which I'll talk about a little bit later. But you can see here that we've broadly funded projects across platforms from DNA, RNA, viral vectors, and protein approaches. And amongst those, Moderna and Oxford AstraZeneca have now been licensed and are being distributed globally in hundreds of millions, if now not billions, of doses of vaccine. There are other advanced candidates such as Novavax and Clover, both of which have demonstrated their phase 3 efficacy, and Novavax now has its first licenses in Indonesia and in the Philippines. So a broad range here, and amongst these, some candidates that are less advanced, but nonetheless have some promise to potentially improvements in terms of vaccines for use later on during the pandemic. Next slide, please. So this next slide, I've already mentioned the early work on MERS and disease X, the portfolio. Specifically with COVID-19, we have put a lot of efforts into harmonizing assessment, so working with NIBSC in the UK and WHO on developing an international standard for use in assays. And we do encourage everybody to use this standard so that assays can be reported in international units rather than in arbitrary units. Tackling variants has been an important piece of the work. So working on assays relating to the variants as they emerge and testing to see if the vaccines have cross-neutralization and to the new variants in a timely manner. The next piece that we are really working on in terms of the pandemic is in relation to broadly protective vaccines. And so we had a call for proposal out for broadly protective beta coronaviruses, and we expect to make some partnership agreements on those in the coming weeks to months. I've already mentioned that access is critical to the work we're doing, and this slide shows you the partnership that we have with Gavi and with WHO, supported by UNICEF, to look at a global fair allocation system to ensure that the world's most vulnerable populations are the first to receive vaccines. And as of today, over 500 million doses have been distributed to 144 participating economies. Next slide, please. So another point in terms of important work, one thing that we've seen through the pandemic, and I think we now have the glory of hindsight with the pandemic, and we know what has been achieved, and it has been quite extraordinary what has been achieved in terms of vaccine development at breakneck speed. One of the things that we have been doing to try and help the vaccine community is to set up what we call SWAT teams and a regulatory advisory group to look at an ongoing basis at the challenges that were being observed through preclinical, clinical development, and with manufacturing. So bringing together both the manufacturers, the developers, the scientists, and the experts in the field to look at the potential bottlenecks in development and to try and find solutions through webinars and workshops specifically. The regulatory advisory group that we chair with WHO, and it includes multiple countries, including the Canadian Regulatory Authority, which gives the regulators as a group to have a look at what the challenges are that are being seen in vaccine development and help to start thinking about the types of guidance that regulators would want to give to the regulators to move their candidates forward. Next slide. And manufacturing is another critical area in terms of working on COVID-19 vaccines. What we were seeing earlier in the year, and I'll talk a little bit more about this in some of the challenges, was that there were some bottlenecks in manufacturing specifically, with a huge ramp up of manufacturing specifically for COVID-19 above what is normally manufactured in the context of vaccines. So we set up a manufacturing task force with industry, with our COVAX partners, to really look at some key areas to work on, which are looking in the short term at optimizing the number of doses that can be produced. So looking at bottlenecks, for example, in input supplies, and beginning to think about how we can have a better globally distributed vaccine manufacturing for the future. So with all of that in mind, and I think it's fair to say it has been extraordinary, the progress that vaccine developers have made, the whole community, the scientific community, the engagement of the regulators. But how can we take the lessons learned here from COVID-19 to think about how to continue to improve our response in the future? So in the next slide, first of all, a few thoughts in terms of vaccine development. I think we can all say that mRNA has been transformational in terms of speed of development like we have not seen before. Usually it would take a decade to develop a vaccine. And the first mRNA vaccines, as well as a vectored vaccine, were developed in under a year, that I think nobody would have believed would have been possible. The quickest time a vaccine had been developed before was 4 years. So this is really quite extraordinary and shows the power of the technology. But we have to recognize that the majority of that manufacturing capacity still remains in a very few countries. And what you have seen from the access to vaccines is the access to vaccines has largely followed where the manufacturing capacity is. So as we need to move forward, we need much more globally distributed manufacturing, and in particular, bringing mRNA to other parts of the world. In terms of vaccine development, there have been many vaccines in the pipeline, but few of these have been from the large multinational companies, largely from biotechs or from academic groups. And getting from a biotech working in early development, an academic group, seeing those vaccines through to licensure need a lot of support. And we need to think of better ways to bring that innovation that comes from those groups together with a much more practical advanced development and moving forward with licensure and bringing partnerships together to make that happen. To move rapidly, we have needed to take risks. It is important that we don't take overt risks on safety and effectiveness. The risks that we are taking are more financial risks. And to give you an example of this, the types of at-risk investments that need to be made to move quickly are in manufacturing, developing a manufacturing process, scaling it up. And we were in a situation in COVID-19 where we were doing this even sometimes before the vaccine was in the clinic, before we really knew whether or not it would work. Normally, you would do it in a sequential approach, but here there was the need to do it in parallel and take that at-risk financing into account while you're gathering safety and effectiveness data that you need. Normally, there's a sequence to animal models through to the clinic. The animal models remain critical. And right at the beginning, there was always a question of, could we, through vaccination, have enhanced disease? And careful evaluation of that was needed. And animal models were critical to show that that was not a risk. But it doesn't mean to say necessarily you can do those in sequence. They are crucial, but you don't have to do everything in advance. Next slide, please. So in this next slide, really just a few words on regulatory. In terms of the engagement of regulators has been extraordinary through COVID-19, and there has been a lot of pragmatism to advance vaccines in development. But we know certain things, again, that can help in the future. If you have a known platform with a large database, that can help you advance very quickly because you have a safety database, you have a view immunologically of how the platform works. So you're just talking about adding an additional, adding an antigen, which has to be evaluated at the same time. And this really helps the regulators review things in an expedited way. Pre-populated regulatory assessment protocols is another important idea, and the need to think about simplified labeling, GMO regulations, batch releases, stability data is absolutely critical. And early discussions with regulators and WHO is vitally important. Next slide, please. So moving to the manufacturing side, we have seen a number of challenges in CMC, raw material supply. We've seen shortages in single-use filters, bags, which have actually even stopped production. Multiple tech transfers have been challenging. We've seen companies transferring their technology while they're still developing it, and you run the risk of having a different product in that situation. The importance of having robust, validated assays is critical. We've seen problems in QA resources, and shipments and import and export of raw materials and vaccine components have hampered the equitable access of vaccines specifically. Next slide, please. So how do we take all of that and think about moving forward? As an organization, although we know that the pandemic is not over yet, we're already thinking about how can we build on everything that we've learned from COVID-19, from other outbreaks, epidemics, pandemics, and be better prepared for the future. And in this context, we look at things in three specific ways. First of all, in terms of an objective to prepare, secondly, transform, and thirdly, connect. And in terms of prepare, one of the key strategic initiatives is to strengthen our defenses against COVID-19 and reduce the risk of coronavirus pandemics in the future. So we continue to fund COVID-19 vaccine development with a specific focus on looking at more broadly protective SARS-CoV-2 vaccines that would have variant proof, would be variant proof. And secondly, looking at broadly protective beta coronaviruses that could protect against both known coronaviruses in humans, but potential future pandemic threats. We also are working on other diseases. I've shown you our priority pathogens, and we will continue to develop those vaccines for those diseases that have a threat of outbreaks on an ongoing basis. But then looking at how do we transform in the future, we are looking at developing virus family vaccine libraries, and I'll explain a little bit more about that in the subsequent slides. But in addition to that, we're also looking at how can we compress timelines for development to 100 days. We have to recognize it's the whole community that need to address these problems. And CEPI really feels the importance of connecting networks of laboratories is critical moving forward, and also to support the efforts of lower middle income countries to make sure that they have ownership of their own national health security plans and activities. Next slide. So maybe a little bit more on the 100 days. This is something that has been taken on, this is our ambition. It's been taken on by the G7, and you'll see in the next slide a little bit more about what we mean by 100 days. This is actually showing you what happened in the COVID-19 pandemic. And you saw actually that Pfizer BioNTech had their first clinical readouts in 300 days after the sequence was available, which was extraordinary. And first use of the vaccine was actually on the 8th of December, 2020, where there were already 68 million cases of COVID-19. So really the question is, what if you could have had those first doses in April when there were only 2.3 million cases of COVID-19? What difference would that have made to the pandemic specifically? And if you move to the next slide, this slide just shows you the situation. I think we're all very focused on COVID-19 and the challenges that that has created for all of us. But we also have to recognize that COVID-19 is not necessarily the worst case scenario. This was produced in the US back in 2006 to look at various different alert levels, how severe a pandemic might be through a pandemic severity index. Actually, COVID-19 is really quite close to the bottom here of this scale. So we do have to prepare for types of pandemics in the future that may be significantly more serious and severe to mankind than COVID-19. Next slide. So with all of this as background, you know, where would we be? Where is the feasibility of a 100-day goal? So what you can see on the left-hand side here is what if you had a high morbidity, mortality virus circulating? How can that impact the vaccination, the vaccine development time? And here it's really about looking at the benefit risk. How much data do you need to have a positive benefit risk assessment for a virus, for the use of a vaccine? And then looking at the right-hand side, do you have the tools to to get to 100 days? Do you have a rapid response platform? Do you have extensive platform data that you already know how that platform works? Do you have the safety data? Do you have the clinical trial networks to move forward very rapidly? And of course, do you have the financing? And obviously, how quickly you can go really depends on how many of those questions you can answer and how well you can answer those questions. Next slide. So with that in mind and looking to the next disease X, this just shows you 4 interrelated strategies that you would need to address to get somewhere close to that 100 days. And the first piece is really early warning systems and understanding the benefit risk assessment of an emerging virus. So we know that people have early warning systems, and it's important that the whole ecosystem develops some triggers for development of vaccines. The work that CEPI is planning to do and others are planning to do as well is work on virus family vaccine libraries. So take the libraries that are the families that are a pandemic threat and look at developing a number of different vaccines approaches for those viruses in those families on mRNA platforms, and do that in advance of a potential pandemic. This all helps towards the 100 days, but also we need to recognize that to use a platform like mRNA, we need to get some prototype vaccines there, need to get other things other than COVID-19 licensed as a vaccine on that platform. Next slide. And this next slide just shows you how you could use this virus family vaccine library work, creating the library and using the information in that library when a new virus hits. You may actually have a match against the newly emerging pathogen and be able to move forward very rapidly as you already have clinical data. You may have a close match, which may be then a first line of defense. Or as we have done with COVID-19, you might have a situation where you have a MERS, something similar to MERS, and a rapid response platform. And you can use all of the information that you have gathered to really hit the ground running and work on developing a vaccine as quickly as possible. Next slide. I mentioned regulatory before, and obviously we can look with the regulators at how to improve response to future pandemics by looking at certain aspects of regulation, streamlining and accelerating vaccine development, adapting guidelines, and harmonizing outbreak readiness pathways. Are all critical areas where regulators can work together on being better prepared in the future. Next slide. And then just a few comments that the importance of the enablers here. I mentioned regulatory. On the next slide, I'll just say a few words on manufacturing. But again, the importance of preclinical models and other aspects to make sure that we are prepared. And just to say that CEPI as an organization is planning to really advance manufacturing solutions to accelerate response to epidemics and pandemics in the future by ensuring that there is scalable manufacturing for low and middle income countries, and also looking from a technological basis at what technology innovations can be used in manufacturing in the future to accelerate development. So really COVID-19, I think we can definitely say we have learned a huge amount from COVID-19, and we should use all of those lessons learned to continue to build our preparedness for future epidemics and pandemics. Thank you.
Thank you. Thank you very much, Melanie, for this excellent presentation. Very informative. And again, COVAX has transformed the face of the pandemic by bringing equitable access of vaccines, in particular to the developing world. So I don't know if I don't have still questions in the chat. Yet, as you know, there is only 53% of people who are currently vaccinated in poor countries. They're even, countries with 5% or even less. So my question is, how, what COVAX can do or it plans to do, to fill that gap? Can COVAX do something? It's only on the hands of COVAX to fill that gap and really make it really equitable. You mentioned several roadblocks, but probably there are others than the ones you indicated.
Yes, I mean, that's a very good question, and it is really very challenging. There have been some delays in vaccines getting to COVAX and COVAX facilities. As we know, vaccine development and manufacturing is very challenging. But some of the challenges we've had is things like borders closing. A large number of doses from India have not got to COVAX, but for a very good reason, that they had a very large and alarming outbreak of the Delta virus. So that is a challenge. So we continue to work to ensure that the doses get to the COVAX facility from our R&D work through procuring with companies, but we're also looking at countries, and countries are making donations, which is great. We would ask that people are transparent about their contracts in terms of bilateral deals, and to think about putting COVAX at the top of the list in terms of priorities. So we continue to work. We are really ramping up towards the end of the year, and early next year we will be getting to our ambition of delivering 2 billion doses of vaccine to the world in a fair allocation system. But it is a challenge, but I think it's really important that we all work together both on the technical aspects, but also on making sure that the doses are prioritized to those countries who are falling behind in terms of vaccination.
Thank you. Thank you very much. I think I have a question from Mark on the chat, or not? No, it's not a question from Mark. So what was yes, the second question was, I noted also that not all companies, so there was Moderna, I saw Moderna. I wonder about other companies and the impact that could have, in particular for projects involved or companies that are involved in this type of projects. And we are talking about regulatory processes, how when these companies are engaged in those projects could have pressure, some kind of pressure to make their products available to this sort of initiative.
Yeah, so as I said, so obviously we moved into R&D very early, and as part of the contracts, we ensured that the developers we're working with give doses to what we didn't know at the beginning what it would be, but what has now become the COVAX facility. And there are many other ways to do that. So, we are engaging with all of the companies that have vaccines that can be used, and developing advanced purchase agreements with them. So this is an ongoing basis, but obviously we were taking a lot of risk at the beginning in terms of funding development, and the return for the risk is obviously in the doses that come out at the end.
Thank you very much for your answer. I do have a very important question, in fact, very interesting question in the chat from Jose Hector Galvez. Thank you for the great presentation. And as the technologies you described reduce the time for development of vaccine, is there any concern about the increase on vaccine hesitancy? Several of those opposed to vaccines cite the short development time.
Yes, and vaccine hesitancy is quite a complex issue. Obviously, vaccines were developed rapidly, but what you can see here is in terms of the intensity of of development, the number of participants in trials, we're talking of 30,000, 35,000, 40,000 subjects being followed and being followed for safety specifically. So there has been very rigorous safety assessment, and that needs to continue after licensure. And with any vaccine, you don't know the full story until it's actually actually rolled out, and the importance of continuing that safety assessment afterwards is critical, and the importance of understanding and listening to what people's concerns are as to whether they want to receive or not a vaccine. So I think it's important that people have access to information that is correct information about the vaccine and the potential limitations, the potential side effects, because all vaccines do have some side effects.
Yes, that's right. So I thank you again very much for this wonderful presentation. And I will take the opportunity to introduce now Dr. Scherer, Steve Scherer. So it is really my great pleasure also to introduce Dr. Scherer, as who is the Chief of Research and holds the Northbridge Chair in Pediatric Research at the Hospital for Sick Children. He's the Director of the University of Toronto McLaughlin Center, as well as the Center for Applied Genomics at Sick Kids. Steve has also had a stellar career in genetics, defining the genetic architecture of many important neurodevelopmental diseases, such as autism. And as you will see in his presentation, he has been also one of our great champions in developing infrastructure and capacity in Canada for translational genomics research. So thank you very much, Steve, for talking us today about the host genome in COVID-19 and future genetic studies in Canada.
Well, thank you so much, Dr. Vidal. If I can just go back to the first slide, please. Thank you. Oh, can you take me back to the first slide, please? The title assigned to me was The Host Genome in COVID-19 and Future Genetic Studies in Canada. And as we heard from Dr. Niemers earlier today, the Canadian government made a significant investment through the Innovation, Science and Economic Development Agency of Canada, or ISED, to study the SARS-CoV-2 viral genome sequence to support the sequencing of 150,000 viral genomes. But also, as we heard from Dr. Musser earlier, to sequence the genomes of 10,000 Canadians who have been infected by the virus itself. And I'll talk more about the second part of the project. And in particular, put my presentation in the frame of capacity building, including for future outbreak and pandemic preparedness. So next slide. And I'll jump right to my summary slide here, the lessons learned from COVID-19. And some of these comments are made a little bit tongue-in-cheek, but I'll provide some context a little bit later through my talk. And the first point is, in a nucleic acid-based crisis, in this case a viral pandemic, the most important thing really is to keep the genomic laboratories open at all costs. And in fact, in Canada, we knew this from the 2003 SARS crisis. It hit Toronto and Vancouver, most predominantly in Canada and of course, Hong Kong. And on the bottom left of the slide is shown the science paper describing the first genome sequence of the SARS coronavirus, led by Mark O'Mara and Stephen Jones' group in British Columbia. And this is one of the nodes of the CGen network that I'll talk about on my next slide. And at the same time in Toronto, and I go back please, and the same time in Toronto, we were developing diagnostic tests for the SARS. Can you go back a slide, please? Thank you. On the right, in the New England Journal paper, we were developing the diagnostic test in our hospital here for the testing in Toronto and sharing the PCR primers, the sequencing reagents and things, actually with Hong Kong and Toronto, because the Genome Center director there had moved Lapchi Choi to the University of Hong Kong. So we had quite a bit of experience around the 2003 crisis, and we knew we had to keep our laboratories open because the SARS-CoV-2 or COVID-19 was going to be even more significant. The second point is the advantage of not being hit first is that you can learn from your national and international colleagues. And we heard this earlier on. Just to remind everyone, Canada is about the size of Europe as a whole, yet it has about half the size of the population of the United Kingdom. We have only 36 million people here distributed across a large part of the country, but actually concentrated within 1 hour of the US border. So there were a lot of lessons learned, and we've heard some incredible talks. We learned much of what was happening scientifically through workshops that Mark Lathrop actually organized on Saturday mornings that many of us continue to join. The third point is your response needs to be proactive, informed, regional, and also national, and it has to evolve. And again, we heard that over and over and over again through all of the talks so far. And in fact, this is the case in Canada too. And what I'll talk a little bit about more towards the end of my presentation is how we need to capitalize on the momentum and move the science and research to the next level. And there's some many incredible lessons learned from John Bell's presentation this morning. And then the fourth point is just a strong reminder that health or disease is really a combination of nature, which is your genome, plus nurture, which is your environment, which is everything that's non-genetic. But in fact, what we've learned from COVID-19 is that your environment can be an invasive virus or an invasive genome. So in fact, your environment can be genetic too, in a way. And I think this is the take-home point is if Canada is going to take health research seriously, it needs ongoing population cohort studies, which include coordinated whole genome sequencing to capture better capture the nature component of contributions to health and disease. And again, we heard that wonderfully from John Bell's talk, and I think we'll hear it in the next presentation also. Next slide, please. So we did have a measure of preparedness in Canada through CGEN, which is the Canada Foundation for Innovation Major Science Initiative. And I'm one of the leaders, along with Mark Lathrop at McGill and Stephen Jones and Mark O'Mara in British Columbia. And these unites the three major public genome sequencing centers. And the vision is to serve as Canada's engine for genomics-enabled research and discovery. And in fact, the mission statement of CFI MSI Awards is to maintain the infrastructure in a prepared state to support research. Okay, so in fact, this was the perfect time to test and kick the tires of this, this national infrastructure. If you go to the next slide, please. In the early days of the pandemic, the first patient diagnosed in Ontario, and I think also this was across Canada, was January 25th. And a lot happened between January 25th and on the top left of this slide, which was March 23rd. Many people were working behind the scenes. We heard Dr. Musser and others working in Quebec. In Ontario and in Toronto, it was March 13th when everything shut down here. And we spent much of the time between the 13th and the 23rd keeping the laboratories open. Okay, so coming back to my earlier slide, we had to keep the genome laboratories open, but in fact, all of the laboratories in our institution across the University of Toronto were told to shut down. And we had success in this announcement that came up from the provincial government of Ontario that had a list of the essential workplaces that were allowed to stay open. And research for the first time in the history of our province was designated as an essential service. So this was a huge success. I actually don't know the status across other provinces, but we were just so happy in Ontario, we could keep things open. We would not have shut down the Genome Center anyways. As I said, other laboratories were sent home on March 13th. And in our building, I'll show you on the next slide, you can move to the next slide. We were the one entity that stayed open in downtown Toronto right through, working 7 days a week through the pandemic. And I just wanted to show this because in particular for our international colleagues, things are different. We heard the word fragmented used several times in Canada. Our healthcare is delivered provincially. And most diagnostic testing is done in hospital-based laboratories. And in fact, I'm at the Hospital for Sick Children. And on the left hand of the slide, we have the largest diagnostic laboratory in Canada in the Children's Hospital here. And then we have the Genome Center as part of CGen on the right-hand side of this hospital. And I point this out, the building on the right-hand side is our research tower, which is 23 stories, and a quarter of the building was paid for by the CFI Research Hospital Fund. And in fact, when we defended the building in our peer-reviewed site visit, the number one answer that we gave back about 10 years ago to the peer review committee was we needed our own standalone building away from the hospital in case we were hit by a pandemic. I remember that quite distinctly when the scientists asked. And in fact, there was an editorial in Science Magazine in 2003 on this. But anyways, what's relevant to this discussion is that on the left-hand side, we ramped up very quickly in collaboration with the diagnostic lab to develop SARS-CoV-2 PCR testing and now do over 10,000 PCR tests as one of the key sites. And there's 11 different sites across Ontario that do the clinical PCR testing. And now the same diagnostic lab is doing clinical viral genome sequencing, over 200 a day, working 7 days a week. On the right-hand side, we focused on supporting the diagnostic laboratory through training, supporting our reagents, staff, technology development, and of course, the host genome sequencing project I'll tell you about now. The last point that's relevant is we are Illumina's biggest account in Canada by far. So we were the one that did the negotiating around pricing for the HostSeq and other projects related and spread those cost benefits, not only within the CGen capacity, but right across Canada. Next slide, please. So this is the HostSeq timeline, and I won't spend a lot of time on this slide, but I wanted to point out on the left-hand side that a lot of coordination happened in the January to March time period. And I know the same discussions, this was happening in Toronto, and Mark and Dr. Musser and others were doing the same in Quebec, and it was also happening in Vancouver. I'll talk about, as I go through the rest of my talk, the study design on the left-hand slide you don't really see. The Genome Canada announcement came in April. And I think the most important point here is that Canada had a mechanism. Our scientists, Mark Lathrop, myself, Naveed Aziz, and others, had a mechanism to talk to the government through Dr. Niemers, who we heard from earlier. And this is also unique, something we didn't have in 2003. We could communicate quite effectively through the scientific leadership with the government, allowing us to actually coordinate this project. I think that was critically important. We'll just keep going to the next slide, please, and I'll touch on some of these items as I go through. Public engagement, we also heard about earlier, and we knew this was going to be a complex project. This is a genetics project. We did not have a lot of infrastructure that touched on all of the fragmentation. We have a lot of expertise, as I'll show you, in collecting clinical cohorts, on sequencing, on doing annotations, but nothing that tied the whole community together right across Canada. And this was one of the articles that we worked with the Globe and Mail, which is Canada's largest, they call themselves a national paper, but on a Saturday, which this article came out, it would reach 2 million Canadian citizens, and put together this educational article, investigative article on the spreading of the COVID virus based on host susceptibility, which was in essence the project. And in fact, it featured a genome scientist shown here, Cameron Chazand, who was a genome scientist in Quebec, but he's now working in Florida. And in fact, he was one of the participants in our personal genome project, where we had already actually sequenced his genome a few years ago and released that data in the public repository. And we were able to study his genetics and use him as a featured individual of the story. What I would say is that this did have significant impact, and I've heard many, many, many people who read the story and enrolled in the study. There was much other outreach. I remember many articles coming out of Quebec at the same time. Next slide, please. This is the HostSeq governance. I mentioned the names. You can just look at the pictures of the names. You'll know all of these people. These are what I call the conscripted willing, as we've heard over and over again. All of this research happened, typically in late hours of the evening after everything else had to get done. We put together a HostSeq study recruitment committee, HostSeq data storage and sharing committee, HostSeq data and epidemiology committee, and of course the whole genome sequencing technical committee. And the individuals are there. We meet every week and are quite rigorous in meeting our milestones. You'll hear a little bit from Bartha Knoppers later. Next slide, please. So the HostSeq workflow, this is nothing novel here. The key was getting the proper consent in place. And as you'll see, this is a project in a minute. This is a project of projects, and getting contracts in place between the participating institutions. One of the lessons to learn here is that Canada is very deficient, at least certainly at our institution and in my experiences in the province here, in having legal lawyers who know how to deal with collaborative agreements between institutions. We typically underpay our legal staff here, and we lost a lot. We lost actually 9 staff through the pandemic at Sick Kids Hospital here. So this was a challenge. We didn't have the people to do the work. The same on the human resource side. And then with respect to the ethics, we actually have a lot of experience. If you go to the next slide, I think it is the next slide. Yes, through the Personal Genome Project and many of our disease sequencing cohort studies, and of course also through the center at McGill, which Dr. Knoppers leads. There's a lot of expertise in developing consents for genetic studies. But I wanted to point out these were the core elements that we insisted on having in all of the participating studies. And I'm just going to read these. And the first is that we knew from the onset we were going to be doing whole genome sequencing. I'll talk about that in a second. So whole genome sequencing had to be specifically mentioned in the consents. International data sharing also. The data had to be used for future genetic studies too and health outcomes. This is forming actually a national repository for future health outcome studies. Commercial use, we wanted as much commercial involvement as possible, actually, who to use this data, explore this data. Sharing of genetic and clinical data through a controlled access mechanism, I'll touch on that in a moment. Storage of the data in a central database, indefinite storage of genetic and clinical data, and not possible to withdraw. It would just be too complicated to do this in a national study. And also, of course, to guarantee low risk re-identification. Next slide, please. So there's now about, I think it's 12 or 13 studies that have now submitted data to HostSeq. I took out my last slide. We have ongoing discussions right across the country with upwards of 27 different projects. These are the ones that have contracts in place that have research ethics protocols and are submitting samples now. You know, there's not a single study design here. We heard from Dr. Musa earlier about the Quebec Biobank design, already contributing data internationally to some very valuable genetic studies. Here at Sick Kids, we're studying pediatric forms of COVID-19, the so-called inflammatory syndrome in kids. In Toronto, we call it MIS-C, and I think in England, they call it PIMS, or pediatric inflammatory syndrome. I won't go through all of the list here, but these are all being collected. They're essentially all the clinical collection and all other aspects of the study are being supported by other grants from, for example, CIHR, Genome Quebec, or other regional efforts, in some cases NIH, and all of the sequencing and informatics are done as part of the CGen HostSeq grant. Now, what I did want to point out here is that one of the major challenges is if you go to the next slide, most of the patients, I'll come to it in a second, that are being collected are from the hospital-based setting. So these are typically severe cases, and many of the PIs leading our studies are actually the doctors who are seeing the patients. So, you know, it was brought up earlier about people writing grants and the whole peer review, how it's slowed things down. In fact, the biggest challenge that we've had is getting clinical data uploaded and the writing of the original grants, because it's the clinicians who are actually seeing the patients at the same time with all of the stresses due to their clinical responsibilities and their home life responsibilities, if they even had it anymore, and everything else going on around COVID. I won't spend a lot of time on this slide, but whole genome sequencing, as I said earlier, we knew this was essential. This is it's an environmental viral disease, as I pointed out earlier. We know there's going to be rare pediatric forms, Mendelian forms. We know there's going to be complex contributors in common variants we saw earlier. And we need to have all classes of variants. In addition, this is going to serve as an initial entree point into a national repository representing, in part, the Canadian population. So anyways, this is what we do well. I would say we do it as well as the UK groups in doing genome annotation. So I'll just move on to the next slide. You can just, the primary study goals here, this slide doesn't present very well on this, but I'll just quickly tell you that the primary study goals from really are I talked about, well, I had it on the earlier slide, but they're based on biomarker studies, biobank studies, longitudinal serology studies, and clinical trial drug responses. And about 75% of the cases are coming from a hospital-based setting, typically are very severe cases, and the remainder are coming now from kind of later round longitudinal studies and also the pediatric forms I showed earlier. Can you go to the next slide? Thank you. Another unique aspect of our work in Canada, there's at least 4 of the projects that are collecting data also on the viral genome sequence that's accompanying the host, accommodating the virus. And they're listed on this slide here. This has been a real challenge in Canada, and I've heard also elsewhere worldwide. We do have the health card numbers, so the health card identification numbers, again, reminding people that for each province, they would have a different card they use and a different database cataloging this information. But I think the next round of data coming will start to connect these, and we've seen some progress on these projects here, at least the ones coming out of the Toronto ones I'm aware of. The idea here is seeing if the viral variant that's being carried has an influence on the host genome variants and in this type of a genes and environment interaction. So there's no data on this yet from our study, but this will come in the next few rounds. What I did want to point out here is that it'll be, I think, really interesting to see if you can start to impute the presence of the variants if you cannot capture that viral sequence data based on time of sample collection and other existing data existing in the hospital sequencing labs, for example. You can just impute which variant it must have been based on what was happening in that local region at that time. Next slide, please. And this is just a summary of the clinical metadata that's being captured around, and this will be open via aggregate data presentations on websites and portals that already exist. The first 1,777 participants here have been analyzed in aggregate, and we have, I think it's upwards of 3,400 or 3,500 full clinical records in the database now. Things have been moving very fast in the last 6 months, and if you go to the next slide, it shows the deeper phenotypic data in the controlled access phenotype database. So I won't spend a lot of time on this, but there's many different types of data going into the database, and we heard some of the other nice biometric data from Dr. Musa earlier. If you go to the next slide. Yeah, so this is the progress on the project. If you count up the total number of samples that aim to be collected through those studies I showed earlier, it's about 13,800. We've now received 7,190 samples. This is up-to-date data. About 6,000 are fully sequenced, and the clinical data fully logged on about 3,500 or so. We're really quite excited and proud that CGen submitted their first data analysis to the October 2021, that's a typo, of the Host Genetics Initiative freeze. And of course, we're now performing our own studies on some of these hypothesis-driven studies I talked about earlier. So you're going to start to see some, I think, very interesting findings flowing very soon. Next slide. The data is available and will continue to be available through a controlled access data, and this is the data access committee that's listed here on this slide. It's led by Professor Man Zawadi at the McGill Center. And I won't go through the composition of the committee, but this is anchored in the infrastructure that Bartha Knoppers has built over the years. Next slide. We're really aiming to get as much data out as possible. And this is just one call. This is a funding call coming out of the what's called the Canadian Statistical Genetics Group, led by Dr. Lisa Strug, offering scholarships, awards to investigators across Canada who want to use the host-seq data in their studies. And there's other local funding sources, and there's a whole network to get this data in combination with other international data into the labs of scientists across Canada. Next slide. And yeah, I wanted to just mention now that one of our most important collaborations with the Canadian Longitude Study of Aging. This is a national cohort study that's following the health of more than 50,000 Canadian men and women recruited from coast to coast between the age groups of 45 and 85. And it's led by Parminder Rain at McMaster University in Hamilton, but it is truly nationwide. And Mark Lathrop's group is doing most of the genetics on this project. The beauty of this is the phenotypes are collected for 50,000 or plus Canadians now. The DNA is collected, and a simple survey was sent out to see those individuals or participants who had been affected by COVID-19 and enrolled in a research study that was consented. And the samples were pulled very quickly from McGill and sent to Toronto actually for sequencing, because we had a contract in place already with the McMaster group. And within a few week period, we actually had genome sequences from the individuals that were affected by COVID-19. So this is the ideal situation where you would have a national cohort. And I think we'll hear about this from the next talk. And if you go to the next slide, actually, I got a little bit of a plug for what we should be doing in Canada. We should have multiple national cohort studies ongoing where we're doing whole genome sequencing. This is the quote is, "If you build it, they will come." This example is the UK Biobank. If we had all of these samples collected already and the infrastructure in place to piece together the fragmentation, we had lots of pieces in place, but we need to kind of bring them together. I think we could have moved much quicker, and certainly going forward with respect to any future pandemics, but actually any human health study, we need to have these cohort studies, including the genome sequence in place. And just, we'll probably hear about it in the next talk or so, but the 200,000 release of the human genome sequences from the UK Biobank that came out, I think, in the last couple of days, I really believe are going to transform and add power to all of our studies. So we start to tease out this relationship of genes and environment. And then my last slide, I just wanted to plant the idea here on my next slide, which is my last one. I became chief of research at the Hospital for Sick Children 4 months ago. And I talked at the beginning of my presentation how the entire laboratories here, which encompass about 2,000 scientists, were sent home in March, except for the Genome Center, which is the 13th floor. And this is plotted out here in gray. If you look at the top, it's pre-pandemic. And then as you go to the right, it's the number of scientists that have come back per day. So actually, we have about 900 wet lab scientists who need the laboratories. They're all back on site now. But in fact, most of our admin people and those who are dry laboratory people are still working remotely. So probably 66% of the capacity of the physical building are actually used in any given time. Most people are working from home. And I think to really come to the question of how will COVID-19 impact research in Canada and what to do, I think is captured by the title of this slide. I think it's going to be very interesting what we do and how it's going to play out in discovery-based science going forward. Because we built models of, you bump into the scientists in the hallway and have the conversation and have your eureka moment. But in fact, now a third, maybe a half of your staff are actually working from home remotely and interacting via video. It's going to be a very interesting time to see how the laboratory develops. So I'll stop there. Thank you so much for allowing me to present. This has been an incredible day.
Thank you so much, Steve, for the excellent presentation. Again, as I said at the beginning, one of the good Canadian examples of how previous investments in infrastructure and excellence allow us to, or allow you to do, to have such an impact. And I have here a question from Marta Crago, which is, this has been a wonderful talk. How do you see genomics fitting into the new Canadian bioscience research funding opportunity?
Well, you know, I think we're talking to the converted here. I think it's Canada does very, very well historically in genetics, kind of studying one gene at a time, models, functional. We really need to move genomics to the population levels. And once we do that, it will impact everything, including the biotech sector. So we heard a very nice answer to a very similar question from John Bell. What's happened also, which is very interesting in the last 18 months, certainly in places like Toronto, Montreal, and Vancouver, I know, is the investment in biotech, in particular from the United States, and venture capital has actually increased significantly. And I've noticed now that all of these new investments around new drug development are using genome in some capacity, either coupling to analyzing models, doing characterization of the CRISPR engineering, sequencing attached to clinical trials. So genomics is embedded in biotech pharma development now. So I think this is Canada's time. And the second thing is we really have to continue to support the training. The three centers I mentioned that are part of CGEN, and there are others, very important ones, are the primary engine for training the workforce who are going out and working in the diagnostic labs in biotech. I mentioned the Sick Kids Clinical Diagnostic Laboratory. I would say half of the staff in that laboratory actually trained in the Genome Center. In fact, our biggest competitor is the diagnostic laboratory. They move recruit our people all the time. But I think this is Canada's time. We're really kind of poised now to take it forward, and it should be seen as a very exciting time.
You mentioned training, and training, at least we were discussing here, training at McGill the other day, and training in bioinformatics, mathematics, and at the same time, biological sciences. So training in interdisciplinary training, how much this needs to be developed, and how much is it developed in Canada, from your point of view? Should this be strengthened?
I think we have the right infrastructure for training, and we have the right trainees. I think we need to increase the capacity for it. If you look at any of the three genome centers I mentioned, probably 40 to 50% of our staff are informatics and people. And when you look at the informatics, they range from hardcore computational people to physicists to statisticians to everything in between. One thing that's worth pointing out is, in particular to the funding agencies and perhaps government people on the line, is the reason the Genome Center concept works really well is scientists want to be close to the data that's being generated now. They need to be close to the infrastructure and technology, but in particular the data. And the best scientists will want to be close to the data that they can see first, because that's how you make a discovery. Right. So that's why it's important to continue to support these centers that are developing the data and training the people to understand it and to translate it out into the broader public sector and private sector economies. So I think we've made a lot of the right decisions in Canada. The government has actually done well. I really truly believe the missing piece is these funded cohort studies, including the genetic sequencing component. That'll push us across the threshold.
Thank you. Thank you very much, Steve. There was another question in the couple of minutes left from Mark Lathrop to Melanie, whether she could give us provide a short overview of how CEPI was funded and how projects, how they decide on which projects support, and how countries like Canada can optimally participate. This is a question for Melanie.
Okay, great. So thank you for that question. So in terms of our funding process, we do fund through open calls for proposals. So they are on our website specifically. So they are around, specific research or development questions, and we invite people to apply according to the eligibility criteria. So the way that we evaluate candidates is through peer review with internal CEPI and external experts. And from that, we fund the project. So we would welcome very much Canadian scientists and developers. We do have some good collaborations in Canada. One of our vaccine developers is VBI, and we also work with Nexcellis in terms of our central laboratory network. So we welcome new partnerships. I think I've explained some of the areas where we're looking to work on in the future. So very much welcome applications. Thank you.
Thank you, Melanie, and thank you, Steve, again. I think we are just on time. We are at 2:10 to end this fantastic session. Thank you for your time. Thank you for the presentations.